Federica GAZZOLA from the University of Pavia and the Aquatic Ecological Lab from University of Pavia. I studied in the lab during my master thesis and now I'm working with the group, with the research team of University of Pavia. We study the following assemblages and we study at a taxonomical level the species that we found in our samples, in our fouling samples. So we have to identify all the species and we study the NIS, the non-indigenous species, and the cryptogenic species and the native ones too. Our studies are very important. We chose three different marinas from San Remo coastal stretch, and we didn't study before. We don't have data for these sites, so it's very important to analyze to study the presence of NIS, of non-indigenous species, and to study how the fouling communities is assemblages, because the presence of NIS in this port and these three different marinas could say to us, to ask to us why we have to study NIS in a harbor and why we have to study very little species, because we study crustaceans, we study bryozoans, bryozoans too, we study serpulids, polychaetes that are very, very, very little and can damage our biodiversity. They can enter in competition with these species, and we have to study and we have to know which species are and why it's so important to study in a new area in which we don't have data for this.